Body weight and fat mass are complex traits that are determined by many dietary, lifestyle, and genetic factors. Genome-wide association studies point to a variety of genetic markers that contribute to inter-individual differences. Among them, a single nucleotide polymorphism located near the fat mass and obesity-associated gene, also called the FTO gene, appears to have the strongest impact. FTO is expressed in the hypothalamus, where it participates in the regulation of appetite. The A allele is associated with increased hunger, diminished satiety, and greater caloric intake, which accounts for the higher mean body mass index observed in large studies. In a sample of 38,759 European men and women from 13 different cohorts, subjects who were homozygous for the A allele had a 1.67-fold increased odds of obesity. On average, they weighed about 6.6 pounds more than those with the TT genotype. The small body weight difference aligns with the well-known fact that no single gene regulates body weight and that other factors play a much bigger role. However, gene-diet interaction studies provide unique insights into what the FTO variant means for personalized nutrition. Several studies show that individuals with the A allele respond better to high-protein diets. A 6-month study of 737 adults found that a diet consisting of 25% of total calories as protein reversed the appetite-stimulating effect of the A allele. Similar findings were reported in a 2-year trial of 742 subjects, which found that the A allele carriers lost more weight than non-carriers on a high-protein diet. 25% of total calories is a good starting point. Other factors, such as body weight and activity level, should also be considered when adjusting dietary protein intake to meet individual needs.